Hello guys, and welcome to another A-level biology video. Mr. Phillips, and I'm moving straight on. Literally I've just recorded the resting potential video, now going on to action potential with you. So if you haven't watched the resting potential, please do, because it'll help you understand this, and I'm not going to recap it very quickly. So effectively, an action potential nerve impulse is a sudden change in charge where it goes from minus 60 millivolts in the resting potential up to potentially about plus 40, and then it drops back down, and that change in charge moves across the nerve, and that then gives the nerve impulse, which then triggers the response. So let's have a look at this. We've not mentioned this very quickly, other than really quickly. So we talked about the sodium potassium pump causing this minus 60 millivolt charge on the inside, which means the inside is slightly negative compared to the outside. Minus 60 millivolts is very, very small. We then talked about this bit here. So we talked about how this happens, and it's because the sodium potassium pumps are closed. Sorry, sodium potassium channels are closed, but the sodium potassium pump is constantly working, pumping two potassium molecules into the neuron, whereas 3 sodium molecules leave the neuron. So just saying there that 2 potassiums go in, 3 sodiums leave, and that gives the outside a slightly negative charge of minus 60 millivolts, and that then is a resting potential. So that's what happens when the neuron isn't being fired. So if we keep going down then and have a look then at the next part, what happens when it's actually fired? So we said the neuron, when it's at rest, you can't see it because of my face, but the pole is called polarized. So minus 60 millivolts is polarized. So now we're going to depolarize the membrane to create action potential. Now, how does this get triggered? Well, the example you're expected to know is either the Pacinian corpuscle, which I'm going to use now, or the rod and cone cells, which have the photo receptor chemicals in there that break down and get bleached, and that can trigger an action potential as well. But this is the one that I'm going to use today, is the Pacinian corpuscle, which, if you remember, we said when you apply pressure, it deforms the layers, the lamellae of the membrane, and that deforms the sodium channels, which allows sodium channels to open, and that means sodium rushes in. And that's stuff that we've done in the previous video, so please look back if you're unsure. And the key thing is this bottom one is that causes sodium gates to open, and that causes sodium to rush in. And if enough sodium rushes in, it triggers an action potential. So it has to get above a minimum threshold for it to trigger the actual nerve impulse, the action potential. So let's have a look then. So basically this sort of shows you what is happening here. So we've got a closed gated sodium channels, okay, and they are voltage gated channels. So basically these channels here, so we're talking just about the sodium ones at the moment, so I've sort of covered mine up annoyingly here, okay, they are closed unless the charge gets to a level of minus 50 millivolts, which is called the threshold potential. So what happens is the sodium channels are forced open, okay, and the potassium channels are forced open as well, but the sodium channels are forced open, okay. That means that sodium can then, if this was open, would be able to rush in to here. Now, if that gets to, let's say, from minus 60 to minus 55, it will just close and there won't be an action potential fired, there won't be an impulse fired. But if it reaches this minus 50 millivolts, so it becomes a bit less negative on the inside of the cell, that then triggers the next sodium channel to open up, and that causes more sodium to rush in, and then as a result that's like a chain reaction then, and that causes the sodium to rush through and move into the channels, okay. And that's the key thing that you need to know. I don't know why I've put sodium there, it's a 2+ charge, okay, so it's Na+. So that's the key thing. It's like a system. Once enough get in, it causes the other sodium channels to open. So sodium comes, and then that triggers more to open. So this is called the all or nothing rule, and basically pain isn't caused, or impulse, like intensity isn't caused by the amount of sodium rushes in, it's the frequency of the nerve impulse. So if you get below minus 50 millivolts, then you get no impulse, okay, it just causes local deformation and it doesn't get to your brain. Above minus 50 millivolts, it only has to be like minus 49, and then that will cause a full action potential, and they're all of the same strength as a result. And basically what happens is there's no halfway. Once it goes past that threshold potential of minus 50, so once it goes above that, say minus 49, then it will trigger the full action potential, and that will go up to about plus 40 millivolts. millivolts as it moves along the atom. Now, this all or nothing thing is important because that means that you don't get like half impacts, you always get full impacts, full action potentials, and your brain detects the frequency of these to work out whether it's pain or pleasure or whatever. So we really need to be able to describe this diagram completely, and this diagram is one that if you think about these numbers, you should be able to say something of each sort of number stage. So if we start off at number 1 here, we're in the resting potential here. So here we're at minus 60 millivolts, okay, which you can see there, okay, and that is because the sodium channels are closed, the potassium channels are closed, okay, and the sodium potassium pump is doing its job actively using ATP to move three sodiums out, and 2 potassiums in. And as you can tell, you always have to double check you get those the right way around. Now what happens then is there's pressure put onto the preceding corpuscle, and at number 2 this causes the sodium channels to deform and sodium rushes into the axon, and that takes the charge up slightly, and if it goes above this minus 50 millivolt threshold, which would be about here on the graph, then it will fire an action potential. If it didn't, what would happen is it would just go up like that, and it would just stay level again and go back to the resting potential. But in this case, we're saying enough sodium has rushed into the cell to cause that minus 50 threshold potential to happen. Now, because you've got enough going in, that means all the sodium channels open, and then we get full depolarization occurring. So at number 3, we're at depolarization of the membrane. It goes above the negative, and it goes all the way up to about plus 40 here, which this one doesn't quite get to, so always read it off the graph. I'd say this is about plus 35, something like that. And at this point, all the sodium's rushed in that can, so then the membrane has to be repolarized. And here what happens is the sodium channels then close when they get to this level of what we call fully depolarized. Okay, so the sodium channels close, and then that causes the potassium channels to open, which then allows potassium to leave. So at this point in the diagram here, and then that takes down the charge back down as a result. Now, what happens though is that too many potassium ions actually leave, okay, and we actually go lower than the original resting potential, and we enter something at number 8 called the refractory period. And basically at this point, no new action potentials can be fired, and we'll talk about the importance of this in a second, but effectively this means that the nerve impulse can only go one way. And it just gives the nerve a bit of moment to stabilise and to basically reorganise its sodium and potassium using the sodium potassium pump, which is continually still pumping 2 potassiums in and 3 sodiums out, which over time, which isn't a lot of time, a few milliseconds, re-establishes the resting potential. Now, the importance of this refractory period is that this, if you imagine going along the neuron, if this potassium channel, sodium channel here, and potassium channel is closed because it's in the refractory period, if a new deformation happens, they can't be triggered. And it means that one can't trigger the next. So effectively, it causes the movement along in one direction. And we'll look at the importance of that when we do about saltatory conduction in the next video. So let's just go through the numbers quickly. Number 1 is a resting potential, minus 60 millivolts, sodium, potassium ions are closed. Number 2, the deformation at the preceding corpuscle happens, that causes the sodium channels to open, sodium rushes in. If it goes past the threshold potential, we then get to number 3 and number 4, where all the sodium channels open and we get depolarization of the membrane. Okay, at number 5, the sodium channels close as you reach the membrane is fully depolarized, and then at number 6, the potassium channels open, so potassium then rushes out at number 7, which is called repolarization. And then then we go to number 8, hyperpolarization in the refractory period, where too many positive ions have left, and therefore it becomes even more negative than the -60 it was before, maybe down to -80 millivolts, let's say. And that is a refractory period where the nerve will not fire another action potential until the charge is restored. Back to number 9, which is when the resting potential is restored due to the sodium-potassium pump again, and the sodium and potassium ions being closed. So you might get different readings of this and be asked to explain it. So you can see here, you can see different examples. You might be asked to explain the difference in charge over time, and you might have to talk through the time of each charge and go through what I just went through there. Or you might have to do something here where it might, instead of saying this, it might say what happens at A. So you'd expect to say at A, the sodium channels open. At B, the sodium channels close. At C, the potassium channels open, and then once it goes to refractory period, the potassium channels also close, and then the sodium potassium pump restores the balance. Really important you learn that, guys, and link it in. So again, I'd go straight on to, if you're happy with that, onto saltatory conduction to maintain your understanding. Thank you very much.